detecting cancer early or, just keeping track of how treatment is working, it's so incredibly important.
Absolutely critical.
And one really promising way to do this, non-invasively, is finding these super rare cells that have escaped a tumor and are floating around in the blood.
CTCs. Circulating tumor cells.
Right, CTCs.
Think of them like tiny scouts almost, carrying intel about the main tumor. Just knowing they're there, how many, what they look like, it tells you a lot.
About how likely the cancer is to spread, the patient's outlook, things like that.
Exactly. Prognosis, potential metastasis, huge clinical value.
But, and this is the big but, right? Finding them is, well, it's notoriously difficult, like finding a specific type of needle in a giant haystack.
That's a very good analogy. They are incredibly outnumbered by normal blood cells. Millions, billions to one sometimes.
So how do we usually try to find them? What are the current ways?
Well, a lot of the existing tech relies on antibodies, molecular hooks, essentially.
Okay, like little flags or something?
Sort of. You might use one antibody that sticks to normal blood cells, like CD45, to get rid of them.
Filter them out?
Yeah. And then maybe another antibody that's supposed to stick to a marker found on tumor cells.
Makes sense. But the research we're looking at today says there's a problem with that.
A big one. This antibody approach, it seems it can miss a lot of CTCs.
Oh.
Yeah, one study they mentioned in lung cancer patients found these methods only picked up something like 40 to 60% of the actual CTCs.
Wow. Wait, so potentially missing almost half of them?
Potentially, yeah. That's a huge gap if you're trying to monitor disease.
Why? Why would they miss so many? If they have the right hook?
That's the key thing. Cancer is incredibly diverse, heterogeneous.
Meaning the cancer cells aren't all identical clones?
Not at all. They can change, they evolve. They might lose the surface marker the antibody's looking for, or maybe that particular cell that broke off never had much of it to begin with.
So your specific antibody hook just doesn't grab onto those cells?
Exactly. You're fishing with a net designed for one specific type of fish, but the cancer is putting out all sorts of different fish. You're inherently biased.
Okay, so the antibody net is leaky because the fish, the CTCs, are too varied. What else is out there? Sequencing.
Sequencing is powerful, definitely. High-throughput sequencing lets you look inside the cells at their DNA, RNA, their molecular secrets.
Which tells you a lot.
It does. Deep genetic insights. But it's not really about finding and identifying individual cells based on how they look in a sample. It's a different tool for a different kind of question.
Got it. So what about just looking at them under a microscope?
Imaging. Yeah, that's a very promising direction because you can see individual cells. You could potentially spot differences in their shape, their size, their structure, their morphology.
Morphology, okay.
But older imaging methods often had this significant drawback.
Which was?
They were often developed or tested using spiked-in cells.
Meaning they added known cancer cells grown in a lab to a normal blood sample.
Exactly. And those lab-grown cell lines, they tend to be very uniform. They've been cultured for ages. They often look quite similar to each other.
Not like the real deal from a patient?
Not really. Actual patient CTCs show a huge range of morphologies. They're messy, diverse. Those neat, uniform lab cells don't capture that reality. So a system trained on them might not work well in a real patient sample.
Plus, didn't some of those older imaging methods still need labels anyway, like fluorescent tags?
Some did, yes, which adds complexity and potential bias again.
Okay, so the picture is, finding rare, diverse CTCs is tough. Antibody methods miss too many, sequencing doesn't visually ID them, and past imaging often use unrealistic models or needed labels.
Which brings us perfectly to the core idea in this research we're diving into.
Right. What if you could skip the labels, skip the antibodies, and just use AI to identify cancer cells based purely on how they look?
Exactly, just their morphology in a standard microscope image.
So the AI looks at a regular bright field image, the kind you get without special stains, and learns to tell cancer cells from normal cells just by their appearance, shape, texture, nucleus.
That's the goal. Using high-content microscopy, really detailed images, and leveraging these huge advances in deep learning.
AI that can see patterns we might miss.
Potentially, yes.
Yeah.
Training AI models to spot cell types and find them in these images without needing those specific labels for the final analysis.
And the potential is not just finding them, but maybe even figuring out where they came from.
That's a really exciting possibility they mentioned. There's some evidence that CTCs from, say, lung cancer might look subtly different from those from liver cancer.
Wow. So the AI could potentially say, this looks like a CTC, and it probably came from the pancreas, just based on visual feature.
That's the hope down the line. predicting tissue of origin from morphology alone. But, and this is crucial, for an AI to do any of this well.
It needs really good data to learn from.
Exactly. Garbage in, garbage out. If you train it on those uniform lab cells, it won't handle the real-world complexity.
So how did this research address that? How did they get realistic data?
They built a new data set using patient-derived organoids.
Organoids. Okay, tell us about those. Why are they better?
Organoids are basically mini organs, or mini tissues, grown in a lob dish directly from a patient's cells.
Like a tiny 3D version of the patient's tissue.
Pretty much. They self-organize in 3D, and because they come straight from the patient, either the tumor or nearby normal tissue, they capture that real-world genetic diversity and, importantly, the phenotypic diversity, how the cells actually look and behave.
Much more realistic than flat cells on plastic.
Way more realistic. So for this study, they generated organoids from 3 patients with cholangiocarcinoma, that's bile duct cancer.
Okay.
And crucially, they used paired samples. They grew cancer organoids and normal tissue organoids from the same patient, from the same type of tissue.
Ah, so the comparison is cancer versus normal, from the same person, same tissue background. That's a much tougher test for the AI.
Very much more robust and relevant benchmark, yeah. Can it tell the difference when the background is so similar?
Makes sense. So how'd they get the images?
They broke down the organoids into single cells, then used high-content microscopy. For each cell, they took 2 types of images simultaneously. Yeah. A standard bright field image, that's the unlabeled one the AI will eventually have to work with.
Okay. And the second?
A fluorescence image. They actually did use fluorescent labels, but wait.
Hold on, I thought the goal was label-free.
It is, for the final AI detection. But the fluorescence was essential for creating the training data. It was the ground truth for the humans doing the annotation.
Ah, okay. Explain that.
They used different fluorescent dyes. Cancer cells were engineered to glow red, normal cells green, and all cell nuclei were stained blue with something called host.
So red for cancer, green for normal, blue for the nucleus.
Right. So the human experts, microscopy specialists, biology students, they looked at the bright field image and the fluorescence image side by side. The fluorescence image told them definitively, okay, this cell in the bright field image is cancer because it's glowing red, or that one's normal because it's green.
I see. So the fluorescence acted like a perfect answer key for the humans, allowing them to accurately label the cells in the bright field images for the AI to learn from. That's clever.
It's a really smart way to generate high-quality training data for a label-free task. They knew exactly which cell was which in the unlabeled image.
And they annotated a lot of cells this way.
Oh yeah, over 75,000 cells across more than 1,200 paired images. They had multiple phases, checks, multiple annotators for the test set. Really careful work.
That sounds like a massive undertaking. And this data set, it's available for others.
It is. They've shared it publicly on Figshare, along with their code on GitHub. That's a huge contribution to the field, letting others build on this.
Definitely. Okay, so they have this fantastic, realistic data set. How did they actually train the AI? What was the structure?
Right. The goal is take only the bright field image and find the cancer cells.
Yeah.
To do this well, especially finding rare things in a busy image, they used a 2-stage pipeline.
2 stages. How does that work for AI vision?
Think of it like a quick scan, followed by a closer look. Stage 1 is a detector network.
Okay.
Its job is to scan the whole image fast and just propose regions, draw little boxes around anything that might be a cancer cell. It's trained to be sensitive, maybe even a bit overly sensitive, to things that look roughly like cancer cells.
So it flags a bunch of potential candidates. What's stage 2?
Stage 2 is a classifier network. It takes each of those little boxes, those image patches proposed by the detector, and examines them much more closely.
Okay.
Its job is to make the final call. Yes, this is a cancer cell, or no, this is actually debris, or this is a normal cell the detector got wrong, or these are overlapping cells.
Why your 2 steps? Why not just one big network?
It helps manage complexity and reduce errors, especially false positives. The detector casts a wide net, finds possibilities. The classifier then carefully inspects each catch. When cells overlap, or there's debris, the first stage might get confused. The second stage, focusing just on that small patch, is better at telling the difference.
Right, like a quick initial sort, then a detailed inspection. Makes sense.
It really helps improve accuracy when you're searching a whole image, not just classifying a perfectly cropped cell.
And they tested this under different conditions, right, to see what information helped the AI most.
They did. 3 main settings. First, just the bright field image. That's the key test for the label-free goal.
Okay.
Second, bright field plus the HOCHIST signal the blue nuclear stain to see if nuclear shape adds useful info.
Right.
And third, bright field plus all the fluorescence, red, green, blue. Not realistic for the final test, but it acts as an upper limit. How well could it do with perfect information? And it also checks the data quality.
Got it. So, the moment of truth. What did the results show? Did the AI actually manage to distinguish cancer cells using only the bright field images?
Okay, so looking at the main metrics F1 score, AURC, which measure how well it separates the classes. For the bright field only setting, at the single cell level, they got an F1 score of about 60.5% and an AURC of 77.5%.
60% F1. What does that mean practically? Is that good, bad? Okay.
It means it's definitely possible. The AI is picking up on visual differences in the unlabeled images. It's learning something meaningful. But it's not perfect. There's still significant confusion. They noted about a 20% mix-up rate between normal and cancer cells in that setting. So it's a proof of concept, promising, but not quite ready for primetime diagnosis on its own, based on that number.
Okay. Proof it can learn from morphology, but needs refinement. What happened when they added the nuclear stain, the HOCHT?
That made a noticeable difference. Performance went up. F1 score jumped to 66%, AROC to 83%.
So including information just about the nucleus shape and texture really helped AI?
It strongly suggests it did, yeah. The AI seems to be effectively using nuclear morphology differences, which lines up with what pathologists have known for a long time. Nuclei often look different in cancer cells. The AI is learning to see that in the stained images.
Interesting. And the perfect information scenario. Bright field plus all the fluorescence?
As expected, performance shot way up. F1 of 94.5%, AUROC over 99%.
Wow, nearly perfect.
Which is really important. It confirms the dataset is good, the annotations are reliable, and the AI model itself is capable of learning this task very well if the signal is clear enough.
So the challenge isn't the AI's learning ability, it's the inherent subtlety of the visual cues in the plain bright field image compared to the obvious fluorescence labels.
Exactly. It's a harder test.
What about finding the cells in the whole image, not just classifying known ones?
Right, the image level detection, that's tougher, more clutter, overlapping cells. The F1 scores there were a bit lower, around 55% for bright field only. But importantly, they showed their two-stage pipeline significantly outperformed just using the detector alone. That second classification stage really helped clean up the results and reduce false positives in the context of the whole messy image.
So that two-stage design proved its worth.
Definitely.
Now, there was that interesting bit about unknown cells, the ones the humans weren't sure about from the fluorescence. How did handling those affect things?
Yeah, that was fascinating. These were ambiguous cells. They tried different strategies. And surprisingly, the best results came when they simply told the AI during training, treat all these unknowns as non-cancer.
That seems weird. Why would calling ambiguous things non-cancer help find actual cancer?
It does sound counterintuitive. But their thinking is maybe many of those unknowns weren't poorly stained cancer cells, but were actually just debris, dead cells, artifacts, things that look different from both healthy cells and viable cancer cells. So by teaching the AI, all this stuff is definitely not what you're looking for. It might have helped the AI learn a more precise definition, a sharper boundary for what a true cancer cell actually looks like morphologically. It's like teaching it to ignore specific types of noise.
That's a really cool insight into dealing with real-world biological messiness in AI training. Okay, what about generalizing? Did it work on patients it hadn't seen before?
That's always the million-dollar question with AI in medicine. How well does it generalize? They tested it on images from patients whose cells weren't in the main training set. And it generalized reasonably well within this specific group of cholangiocarcinoma patients. The performance drop, the F1 score, was less than 2% when moving to unseen patients from this cohort.
That's pretty good, suggesting the morphological features it learned weren't totally unique to just the training patients.
That's promising, yeah. But they rightly caution this doesn't automatically mean it would work perfectly on, say, a different type of cancer or patients from a completely different hospital system or population. Generalization is complex.
Still, a good sign for this cancer type.
And they suggest their model could be a great starting point for transfer learning. Other labs could take this pre-trained model and fine-tune it on their own local data.
Right, leveraging their work. Did dataset size matter? More data equals better AI.
Absolutely.
Yeah.
They showed a clear trend as they increased the amount of training data, especially for the realistic bright field and bright field plus HOCHT inputs. The performance kept going up. The curve hadn't flattened.
So more data would likely mean even better results.
Strongly suggests that yes, more data capturing more variations would probably push the accuracy higher.
Okay, so let's zoom out. What's the big takeaway here? What did they really achieve?
I'd say two main things. First, they created this incredibly valuable, large, realistic data set using organoids and made it public. That's a huge resource.
Right.
Second, they provided a solid proof of concept. They showed it is feasible to use deep learning to classify cancer cells based purely on morphology in unlabeled images. They highlighted the role of nuclear features and the benefit of that two-stage pipeline for dealing with messy images.
So they've laid some important groundwork. What's the potential impact if this approach matures?
The potential is really significant. Imagine an automated system for finding and maybe even characterizing CTCs that doesn't depend on specific antibodies.
Which could mean it works across more cancer types and catches those diverse cells the antibodies miss.
Potentially, yes. More universal, less biased detection method.
And where does this go next? What are the future directions?
Well, the obvious next step is more data. Lots more data. More patients, more cancer types, different tissues, different disease stages. Really capture the full spectrum of morphological diversity.
Build an even bigger, better library for the AI to learn from.
Exactly. And then the really exciting long-term vision is linking morphology to outcomes.
You mean, could the AI look at the shape of a CTC and predict something about the patient?
Potentially. Could specific morphological features predict disease progression, or how a patient might respond to treatment A versus treatment B, using the visual information as a biomarker itself?
That would be revolutionary. Getting prognostic clues just from how a cell looks through the AI's eyes.
That's the ultimate goal, finding those hidden visual patterns. Of course, there's still big challenges getting enough data, making it robust to artifacts in real clinical samples. But this work is a major step, and making the data and code open source really helps the whole community move forward.
Yeah, this deep dive really highlights how combining these realistic biological models, like organoids, with powerful AI is opening doors we couldn't before, tackling that old, hard problem of finding CTCs in a new way.
It's pushing the envelope on what we can learn just by looking very intelligently at microscope images.
So think about this. If AI can learn to spot cancer by detecting subtle visual differences in cells, differences we might not even consciously notice just from a standard microscope image, what other hidden clues are lurking in biological images? What else could AI learn to see for diagnostics or just understanding disease? How might this change screening or monitoring in the future? Could it make tests faster, cheaper, maybe less invasive, just by getting smarter about analyzing the pictures we can already take?